So great. Warm welcome from Bolzano, from the ECHO team, to this webinar on soil biodiversity data in practice. Today we will hear three presentations about interpreting the results from the ECHOREPO. You've been really active. I just checked our ECHOREPO this morning. We are over 5,200 input soil data on our database. So a big thanks goes to our citizens out there across Europe. So you're doing a great job. And of course, increasing the number of samples means a lot of data, also biodiversity data. And we got a lot of questions from you to how to interpret and what's the data about. So that's why we organized this webinar. We have three speakers today. I will briefly introduce them right now and then give the floor to them. So we have in first place Ilaria Fracasso from our university. She's a postdoc at our university. She has a master in biotechnology for health and environment from the University of Ferrara, a PhD at the University of Bolzano. And now since a couple of years, she's with us working on biodiversity in different ecosystem, agriculture, alpine, natural ecosystems. Our second speaker, and of course I did not say, a big thanks goes to them giving the availability to speak today. Alberto Orgiazzi from the GSC, the Joint Research Commission. He's also an expert. I mean, all the first two speakers are experts on biodiversity. He has a master's degree in plant biotechnology from the University of Turin and a PhD in biology and biotechnology of fungi from the same university. And he's now, as I said, working at the GSC, and he's expert research consultant to the European Commissions. And his research focuses on investigating soil biodiversity by means of molecular biology approaches. Our last speakers, I'm sorry if I pronounce it or mispronounce your name, Oleg Olshenko. I know it's totally wrong, but I will work on that. He's not a biodiversity expert, but he's a data expert and IT expert, and he's also a partner of our project. And he's actually the backside of ECHOREPO because he's the big one dealing with all the issues and all the setting up of the ECHOREPO. So a great big thank goes to Oleg. He has a PhD, just some background information from Oleg. He holds a PhD in computational and applied physics from the Universitat Politècnica de Catalunya. And he's currently a data science a scientist at Quanta Labs. So I won't take too much time because I think the talks and the presentation are much more interesting than my presentation and my talk. So I give the floor to Ilaria. Thanks very much. And I'm really curious to hear your presentation.
Thank you, Tanya. Okay, let me share my screen. Can you see my presentation?
Yes. Yes.
Okay.
Thanks.
Thank you, Tanya. So I'm Ilaria Fracasso, and today I'm here to talk about soil biodiversity. And I would like to start with a question. I think that you may have a garden or a park nearby your houses, or just a pot of soil on your balcony. But I'm also quite sure that you walk right past them without giving them a second thought. But what about if I told you that in soil you can have more living organisms than the entire being on human being on our planet? And this is not just a metaphor, but it is not an exaggeration, but it's the reality we're here to talk about today. So first of all, I would like to, I'm sorry, I'm not able to go. Okay. Okay, so sorry. So I would like to start with soil. So what is soil? I think that for most people, soil is just something dirt, but in reality, soil is three things at once. Soil is physical structure because it is represented by an architecture formed by different minerals, by sand, by silt, and by clay. But soil is also a chemical system because it is rich in nutrients, and all the ingredients that plant and other organisms need to grow. And it's a biological community because soil is rich in a lot of living beings like bacteria, fungi, nematode, and other insects, and they all together form a huge underground city. And to understand the full picture of soil, we need all these three lenses. But why biodiversity is important? Why biodiversity matter? Because soil is not just the place in which plants used to grow, but it's the substrate we use to grow our own food. It's the biggest water filter present on Earth, and it is also able to store an amount of carbon much more higher than the total carbon that all the forests combined are able to store. But of course, soil is not able to do that alone, but the real actors able to do to perform all these functions are the organisms in soil, because they are able to cycle nutrients, they are able to filter pollutants from groundwater, and they are able to regulate the storage and release of carbon in soil under different climatic scenario. So that's why organisms are important. And what I would like to present you is the different organisms living in the soil, and they can be divided in different layers. We have the microscopic layers, which include bacteria, fungi, protozoa, and archaea, and they represent the base of this soil food web. Then we have an intermediate level which is represented by mesofauna like nematodes or mite, which include organisms that used to graze, for example, bacteria and fungi. And finally, we have the macrofauna, which include the well-known earthworms, which are the classic main engineers in soil. But in this project, we focused mainly on bacteria and fungi because they are the most abundant in soil. And as I said before, they represent the base of this soil food web. So first of all, bacteria are the microscopic engineers of soil. They are a lot. Think about that in a single teaspoon of soil, we can have billions of bacteria, and they are divided in different groups, and each group has its own function or his own functions. For example, bacteria can convert nitrogen into plant available forms. They are decomposers, so they can break down organic matters, and they can also act as biocontrol agents. In some cases, they cause plant disease. In some other cases, they protect plant against disease. Secondly, we have fungi. They are even more interesting than bacteria because think about again that in a teaspoon of soil you can have 200 meters of fungal threads, and they can form an underground web, which is called by some scientists the wood wide web, and they are able to connect all together different plants, and they are able to transport like nutrients, water, but also signals. And their role is very important because in some environments they are the only organisms able to break down the woody materials. But how we studied the biodiversity and how you can read your results. So first of all, we studied these organisms using DNA. DNA is a molecule which is present in all organisms, both living organisms and dead organisms. And to study them, we selected a short section of this molecule, which is called barcode. Barcode, it's like a biological passport. When you have a passport, you are able to learn the name of a person and all its characteristics. A barcode is exactly the same. By reading this barcode, we can learn the name of the organism and also all his characteristic. And we selected the 16S rRNA for bacteria and the ITS for fungi. But the number of barcode is very huge, and we have many different barcodes and that can be used to study many different organisms. For example, the CO1 for macrofauna or the 18S rRNA for protists and algae. And by studying these barcodes, we were able to produce a list of organisms of the organisms that are present in your samples. And the first chart that you can find on the eco report is the taxonomic chart. What I would like to highlight is that in this chart we decided to represent only the identified organisms, because of course at the moment we don't have information for all the bacteria and all the fungi present on our planet. So some of them are still unclassified, unidentified. And we decided to present here just the identified organisms, so only the organisms for which we have some information. And in each chart, you can see the 10 most abundant family or phylum per sample, and each slice represents a specific organism. The dimension of the slice is connected to the amount of DNA that matched that organisms. But what I would like to underline is that the dimension of the slice cannot be directly connected to the importance of the role of the organisms in the soil. So if the slice is big, it doesn't mean directly that the role of the organisms in soil is more important. And when we had produced this list of organisms, we tried to give to each organism a specific role, and we were able to establish the potential functionality of these organisms in your soil. Why I'm talking about potential functionality and non-real functionality? Because as I said before, with the DNA, we can find both living and dead organisms. And of course, it's like that with the DNA is like a photos of a person, because if you see a person that is doing like this, you can say, okay, maybe he's saying hello to someone, but without a film, without a video, you cannot confirm that he's really say hello to someone. I don't know if I explain this concept properly, but if you have question, I'm here to answer. So by using the DNA, we were able to establish the potential functions of your communities, and all these functions were grouped in bacterial and fungal ecological guilds. So when you read your graphs, you can say, for example, that in this case, the 70.7% of the community of the identified community in these samples is represented by aerobic heterotrophs, which means that the 70.7% of the identified community is potentially able to degrade organic matter in the presence of oxygen. And don't worry, because we are going to provide also a file that will help you to understand what aerobic heterotroph means, what anaerobic heterotroph means, and so on. And what I would like to underline also for regarding this graph specifically for fungi is that you may find something like plant pathogens and animal pathogens, but you don't have to worry about this data, because they don't mean that your soil is dangerous, because the detection of a specific function cannot be directly related to the presence of the disease. First of all, because bacteria or fungi need specific condition to be able to cause a specific disease. And secondly, because, as I said before, the DNA is from both living and dead organisms. So don't worry about these results. So this is a picture of how you can read your data, but if you need more information, you have my email in the first slide. And I would like to conclude with an answer. The question in my first slide was who is living there. So nearby us, an enormous amount of life is living, and this life is represented by communities that are working every day silently and invisibly to feed us, to protect us, and they are trying their best to maintain our planet stable. So thank you for your attention, and thank you for your interest in soil science.
Great. Thanks very much, Ilaria. And I think in the meantime, while we switch to the next presentation, maybe Alberto is going to share the screen. And to all those that are listening, you know that you can contact us anytime, or the speaker directly, or us via contact ECHO or me as coordinator. So feel free to contact us anytime. So in the meantime, also the sharing is working. So Alberto, the floor is yours.
Yeah. Good morning. I hope you see my screen. Yeah, thanks for the invite. As said before, I'm Alberto Giazzi, and I'm currently working as a research consultant to the Joint Research Center, which is the scientific branch of the European Commission. So we provide science to policymakers for taking decision on different type of policy, environmental, climate, agricultural policies. So I'm going to talk about what we do at the JRC on soil biodiversity, starting from this picture, which is not that good. The message is clear. Back in 2016, we tried to develop maps of potential threats to soil biodiversity across Europe, taking into account different type of risk like soil erosion, pollution, climate change, and so on. And as you can see, most of the regions in Europe are not doing well for soil biodiversity. For this reason, there is a strong need to monitor soil biodiversity to understand how is the biological condition of European soils. But it's not that easy, this task, because we don't know that much about soil biodiversity. Most of the species that live in the soil are still unknown, have not been described compared to what is living above ground, where we have indeed more data. We have indicator species. We have a standard methodology to measure biodiversity. All this is not present for life in the soil. So it's a very big challenge to do this, to monitor the organisms living in the soil. But however, as said before by Ilaria, over the last year, there has been a strong development in research thanks to DNA molecular biology technique, the use of DNA barcodes, so the possibility to extract DNA from the environment, from a soil sample, that is composed by the DNA of all the organisms living in that sample. So if we are able then to sequence the DNA, we have a clear picture of the organisms, of the biodiversity living in that soil, so we are able to describe the biodiversity. And in this context, came the Lucas Soil Project, which is the largest European soil survey coordinated by the JRC, the Joint Research Center of the European Commission. Everything started in 2009, where we started collecting sample, as you can see all the points, the yellow points, across Europe, for analyzing at the beginning mainly physicochemical properties. All the samples are collected in the same standard way, and also analyzed in the same standard way, so that they can be easily compared. And the good point of this project is that it has the possibility to grow over the years. So as I said, we started in 2009 analyzing mainly physicochemical properties like carbon, pH, nitrogen, phosphorus, and so on. But as of 2018, we were also able to add new components, like, for instance, pesticides analysis in the soil. But for us, it's more interesting also the possibility to include biodiversity. And as I said, we decided to use a molecular biology approach, so DNA, The idea was to collect, of course, was not possible to collect soil samples for soil biodiversity analysis in all the points that I showed before, because it's complex, the analysis and the collection of samples for soil biodiversity. But at the end, as you can see, our freezers packed with soil frozen samples. We were able to send kits for collecting fresh soil samples with boxes and ice packs, and that has allowed to generate what is the largest so far soil DNA datasets across Europe. And how we have used this, we are using this. Back in 2021, the European Commission launched the EU Soil Strategy for 2030. And among the activities on related to soil, there was one on soil biodiversity, and specifically the production, the development of the first assessment of the soil genetic diversity in Europe. So we used those data collected in 2018 to do this, to perform this first ever genetic assessment of soil biodiversity across Europe. As you can see, we were able to publish this. Mainly two papers, one focused on microbes and one focused on, let's say, animals, soil animals. And what we found, I'm going to show you some interesting results related specifically to microbes, so bacteria and fungi. One of the most interesting things that we found is that the more intensively used lands, like in particular croplands, so agricultural soils, were those that have higher diversity number, let's say number of species. And this may be surprising to us, but it's not if you think that soils, agricultural soils, are soils that keep on receiving inputs from like fertilizer, but also pesticides that for microbes can be a source of energy, let's say food. So this can be one of the reasons why in these let's say artificial soils there are more diversity. But we not only focus on the taxonomical diversity, so the number of species, but also on the functions. As said before by Ilaria, it's also important to understand what those organisms do or potentially do in the soil. And that's another interesting thing that we found is that, for instance, of course, agricultural soil, they have more diversity, but there are also more pathogens. So let's say more dangerous, let's call it like this, dangerous organisms. So if you want to have a clear picture of the biodiversity in a soil, it's not just important to understand where there is more diversity, but also to focus on the functional aspects that can tell you something different, opening also kind of philosophical question, because if we want to protect biodiversity, no matter what it does, we should also protect pathogens, because in a way they are part of the ecosystem. But if we are focused on humans, then pathogens may be dangerous, so we should not protect them. As I said, we not only measured biodiversity, but also other properties in the soil, other elements, and in particular pesticides. Residues were analyzed in 2018, and that has allowed to combine these two data sets, pesticides and biodiversity, with very interesting results that were recently published in a paper in Nature that also got the cover, as you can see. And what we found, combining pesticide and biodiversity data at European scale, first of all, we found that pesticides are present in all soils, not just the agricultural soil, but also more natural soils like grasslands and woodlands. We also found that pesticides are not always dangerous to soil organisms, and that is not surprising. Again, if you think of what I said before, that pesticide, fertilizer can be energy for microbes. So in a way, some group of organisms can use pesticides to grow. But what we found is that back to the functional aspect, that some of the beneficial organisms like arbuscular fungi, which are symbiotic fungi that help plant growing, are negatively affected by the presence of pesticides. So once again, the importance not just to focus on taxonomy, but also on the functional aspect. That is what we have done so far. We are, of course, still working on this Lucas soil biodiversity data. And in particular, we are now trying to map biodiversity at European scale. Mapping is a key component for monitoring. If you want to protect something, you need to know where this something is. And so far, there are not maps, high-resolution maps of biodiversity at European scale. So thanks to this, Lucas, we will be able, hopefully, to fill in this gap and then identify hotspots, soils, regions rich in biodiversity that can then subjected to action for specific action for protection. Of course, all this is not easy. While Lucas soil biodiversity is a good example, but I would say it's an exception. There are not many soil biodiversity monitoring schemes at national and regional scale. Monitoring soil biodiversity for all the things that I said before is not easy. Also, using molecular biology is not easy because there are many steps that need harmonization. There are different methodology for extracting DNA, for analyzing the data, the DNA sequencing. So there is a lot of discussion within the scientific community to try to harmonize all these steps. We also have to remember that soil biodiversity is very complex. I show you results on microbes, but soil biodiversity is not just microbes. There are many more organisms living in the soil. Most of them are still unknown, so it's impossible to monitor them. The good news is that as of December 2025, the European Commission has adopted a legislation aiming to protect soil and its biodiversity. It's so-called the Soil Monitoring and Resilience Directive. Indeed, in Europe, so far we only had legislation to protect water and air. Soil, this important environment, was missing. Now we have legislation for this, and also soil biodiversity is included among the parameters that should be monitored. As you can see in the list, there is actually only one mandatory parameter that should be measured by member states, which is the DNA metabarcoding of bacteria and fungi. But then there is a long list also of optional indicators. Of course, it's not probably the best, but it's a starting point. It's the first time that soil biodiversity is into legislation, and there will be in the future the possibility to improve and expand this framework. But of course, this is something more bigger than our daily life. It's related to national Ministry of Environment, and then there is us in everyday life what we can do on soil biodiversity. I think the first important thing is to be aware there is life in the soil. Life in the soil is important, and to be aware, the most easy thing to do is try to learn, study more. And in this regard, the Joint Research Center in the past has produced an important tool, the Global Soil Biodiversity Atlas, that was published in 2016. There are a lot of information there, pictures, images showing also the beauty of soil biodiversity. You can download this. It's free. You have the link there. But if you Google the Global Soil Biodiversity Atlas, you find the link. And then, of course, there is important initiatives like ECHO project doing citizen science. Not just the Atlas. We also developed in the past another important tool, this online course that you can find in the EU Academy platform. Again, you just Google the name. It's a free online course, not just for teachers and students, but everyone can enroll and take look at videos, watching videos, taking quiz activities to learn again on the importance and relevance of soil biodiversity. And that's it. Here are the link to the EU Observatory, Soil Observatory, where you can find all the data, Lucas, soil biodiversity data, information, and then my contact in case you have questions. Thank you so much.
Well, thanks very much, Alberto. It was a great presentation. It's always really nice what you're doing there. And then also the Atlas, it's great. I have it actually the printed version in my office, and it's always really great, simply great, just, looking at the great pictures and the content. So maybe in the meantime, Oleg, you're ready. Sorry, I did mispronounce again your name. And now we heard a lot about biodiversity, and now we will be hearing where are our data and where we're getting to this data, and maybe some more details about our ECHOREPO. So the floor is yours.
Just a second. I'll share the screen. Yes. So yes. Hello. My name is Oleg Kosachenko, and I represent Quanta Labs, which is a partner that is providing the data solution for the ECHO project. And the data solution is actually ECHOREPO, our repository. I will say a few words about the mobile application. Mobile application is kind of out of scope of this presentation, but this is the cornerstone of how the user, the citizen scientist interacts with the database and how they are supposed to work. So a user installs the mobile application on their phone, takes the sample, and the mobile application gathers all the necessary parameters, physical, chemical parameters, and other parameters, and it makes sure this data reaches the database correctly. And let me just make a live presentation of how, after all, a user, a person, a scientist, or a contributor can take a look and can download data and consult data. So first of all, this is our host. And when I enter here, so this is the landing page. We see the map that is representing all the samples. You see the color coding in accordance with the pH. And also we can take a look and see, for example, at some samples. So you see the basic information regarding this sample, but in the current implementation, we only can consult single data for the samples, and not to download it unless we are logged in. And we log in with the same account that is created in the mobile application, and I will do it right here. And here, once you are logged in, you can reach and make use of all the functionality that ECHOREPO offers. So first of all, you may see that we have a number of languages available for the website, and all the materials are translated to the corresponding languages. Then I will later I will take a look and explain what these buttons mean. But first of all, let's take a look at the map itself. So like I said, we have the representation of all the samples currently available in the database. And a person might, for example, take a look at some kind of subset, export, download the data for this subset, and also download the whole database. The database consists of 4 basic basic files and basic tables. So the first one is the actual samples gathered by the scientists. Then the references to the images gathered by these citizen scientists, and two other tables are the raw data for the biodiversity and the raw data for the elemental concentrations, for the laboratory analysis of the elemental concentrations of those samples. These are for metals and non-metals alike. And beneath the map, we can take a look at the samples assigned to us. So these are actually testing not real data, but some kind of test samples. And you can see, for example, one of the samples has the elementary concentration information, for example, and also the images. You may consult the images assigned to these samples, and you also can take a look at each sample separately. So I showed you the how this pop-up works. And now let's take a look at the search. So in the search, you can perform a more granular selection of what you are interested in. For example, you might be interested in particular codes, for example, or you might be interested in a particular country, or you might be interested to a particular range of pH or the range of dates where the sample was taken. For example, it might be winter or it might be summer. And then you might export the results of the search. We get back to the initial page. Then for if a user is has samples in the database, and these samples also contain information about the elementary concentrations, they are offered to actually fill up the final survey. So this is the link that leads you to the external survey that holds the survey. Also, ECHOREPO is capable of analyzing actually the possible inconsistencies in the locations of the samples, and allows, for example, if some kind of error is identified, allows the user to correct it in case, for example, they introduced some kind of wrong coordinates. You might, if you know the correct coordinates, of course, you might put it here and correct it. Then all the exports, let me download just one file, and let's take a look at it. It's a CSV file in more or less standard naming of the columns, pretty self-explanatory. But what is important here is that the data set already has a citable reference, since our data sets are mirrored on Zenodo. So these are the permanent identifiers that might be used to cite this dataset. And ECHOREPO also takes care of synchronizing periodically the database with the Zenodo storage. Then, like I said, the map allows you to perform selections and just pick something, just maybe select a chunk of a country, or maybe something that is interesting to you, for you. And also it allows to just show you the samples that are yours, so to say, the samples that are taken by you. And let's get back to the search. And just one last element that might be interesting to you. I will look for just more or less random sample, and let's take a look at it. Sometimes it takes several seconds to load. Just a moment. Yes. So I took this one in particular because this sample contains data for the biodiversity analysis, and this is how you might actually assess this, actually in accordance with what Ilaria explained before. So on the bottom of the pop-up, you might find all these four graphs that were shown regarding the analysis for 16S and ITS barcodes for bacteria and fungi, and also for ecological gifts. So, for example, if you take a look, this is, for example, response to this sample. These are, of course, downloadable, and they are generated automatically based on the raw data available for this sample. And last element of ECHOREPO that I wanted to explain is actually the API. You are very welcome to our GitHub, which is public, open to everybody. And in the documentation of the repository, you might find actually all the documentation regarding the development. But I wanted to explain a little bit more about the API. So here, the ways of machine-to-machine interaction with ECHOREPO are explained just step by step. The only thing needed for this to work is actually only the API key, which is provided on a personal basis by us. And this is already working. So if you need an automatic interaction and connection with ECHOREPO, and it has a pretty rich amount of functionality regarding the search, filtering. So you might consult our repository and find all the details. So thank you very much.
Great, thanks, Oleg. So now you got a lot of more details about what's ECHOREPO and where you can get the data. And I hope you will have the chance and you have the time and dig into your data, also the data of other citizens across Europe. With this, I thank again the speakers. We had really great, very, very informative presentations. A big thanks goes to all the three of you. With this, I close the webinar. Thanks again, because a special thanks goes to the citizens out there. So without you, we could not do this project. So again, thanks, and thanks for listening, and see you soon and talk to you soon. Bye-bye.
Goodbye.